Okay, good afternoon, ladies and gentlemen. My name is Paul Gisson. I'm a professor at metabolic medicine at the Great Ormond Street Institute of Child Health and Great Ormond Street Hospital. And it's my great pleasure this afternoon to present the lunch hour lecture on developing therapies for muscular dystrophy and spinal muscular atrophy, which is given by Professor Francesco Muntoni. Francesco Muntoni is a professor of pediatric neurology and the director of the Dubowitz Neuromuscular Center at the UCL Great Ormond Street Institute of Child Health and Great Ormond Street Hospital for Children. He trained in Italy, but has been working in the UK since 1993. His interest is in pathogenesis, deep phenotyping, gene identification for rare neuromuscular conditions, and also, importantly, translational research in DMDs, spinal muscular atrophy, and congenital myopathies. His research, funded by the Department of Health, MRC, and the European Commission, led to the development and early clinical trials of 2 antisense oligonucleotides, which are now approved by FDA, that induce partial correction of the processing defect of the DMD gene in boys with Duchenne muscular dystrophy. He is the UK chief investigator for many clinical trials on DMD and SMA, which do include AAV trials. He is theme lead in the novel therapies at the Gosh Biomedical Research Center and previously led developmental neuroscience program. Francesco is a co-director of the MRC International Center for Genomic Medicine in Neuromuscular Diseases. And very excitingly, his new role is as a director for Gene Therapy Translational Research Partnership between Institute of Child Health and Neurology, a genetic therapy accelerator center. This center will combine laboratory and clinical expertise focused on neurological, neuromuscular, and neurodegenerative disorders, their mechanism of disease, and cutting-edge RNA and AAV gene therapy expertise, and foster cross-institutional collaborations between Queen Square Institute of Neurology and the Institute of Child Health. Thank you very much for joining us, and over to Francesco now for his lecture.
Firstly, Paul, thank you very much for your kind introduction. We work together in the Biomedical Research Center for now several years, and I really enjoy working with you. I also need to share my screen and give you my presentation of the work that our group is doing in the field, as indicated by Paul. I think that I cannot underemphasize how exciting and how different my job is today compared to 5 years ago. And I think I need you to hopefully get enthusiastic how things are changing our eyes. That's not only neuromuscular disorder, of course, but in many other conditions. This will be my disclosure. You will have them in the presentation. I do not have any direct revenue for any of the medication I will be discussing. But of course, I do work with industry to develop novel therapies. And I will explain and take you through 2 different concepts through my presentation. And these 2 concepts apply well both to Duchenne muscular dystrophy and to spinal muscular atrophy. Of course, I will explain to you what these conditions are, but I will discuss firstly the role and the impact that dealing with mutant RNA has, because there are very interesting development for both conditions dealing with this. And briefly, I will also discuss where the field is moving, displacing genes with gene therapy, as Paul mentioned, are also involved in industry-sponsored research, but this led to 3 treatments also available in this country for our children, and we have been actively involved in that work. So just very quickly, what is Duchenne muscular dystrophy? This is a condition only affects boys, is an X-linked condition. It does affect every year in this country, approximately 80 children are born with a condition. It does lead to progressive loss of muscle and destruction of muscle, to the point that eventually by the age of 9, 10, 11, 12, children stop walking, and eventually they develop cardiorespiratory difficulties, and without intervention, survival is really not possible past the middle 20s and so on. And why they develop this terrible condition is that because in the muscle fiber, this is the sarcolemma, this is the sulfase protein, the sulfase of the muscle cells. One particular key protein, this yellow protein that really works almost as a shock absorber, is absent. And without this protein, that here you can see in these nice red rims as a circular rim again around each single fiber, children with Duchenne completely lose it, and eventually they lose muscle. And this is what a muscle biopsy of a late ambulant child looks like. You can see a few muscle fibers in a sea of connective tissue and fat. And that, of course, is why these children become weaker. I think for those colleagues, for those people who do not have experiencing Duchenne, this is a young child with Duchenne on the left-hand side that is running with already a little of difficulty. This is a presentation. This is the first visit of a child. Then ambulation becomes increasingly difficult because of progressive weakness and contractures. There is like tightening of the Achilles tendon, and then eventually walking becomes impossible. And this is a child who can really only do, as you can appreciate, a few steps before needing to rest. And eventually these children will lose the ability to walk. Now, is an X-linked condition. I did not say, but perhaps it's useful to know that one of the reasons why it continues to occur is because there is a high rate of de novo mutation in the population. And these de novo mutations are very often out-of-frame deletions, some a duplication, then there can be some nonsense mutation and some small mutation. These are the 79 exons of the dystrophin gene. And you realize that is different color code, because if you take, for example, exon 3, this means that blue and square means that if you remove exon 3, you're removing an integral number of nucleotide, and therefore 2 and 4 can stick together, and the protein looks identical with the exception of missing the information encoded by exon 3. While if you remove exon 6 as an example, then 7 and 5 do not match together. And this leads to an out-of-frame deletion, for example, and the an inability to produce dystrophin. Now, nature already gave us some indication that, that there might be the possibility to work on the mutant gene and trim it to in a way to make the gene able to produce at least some protein and lead to a better phenotype. And this is demonstrated in nature by the condition that is called Becker muscular dystrophy, that is a much milder form of muscular dystrophy, where most people remain able to walk for life, where you can see here in this slide, there is the production of some protein. And I think what is interesting in people with Becker is that they also have deletion in the dystrophin gene, but they have in-frame deletion instead of out-of-frame deletion. So one line of thought in the field is how can you trim and affect messenger RNA, splicing of the pre-messenger RNA, and make sure that the Duchenne mutation behave as Becker-like mutation. And I have here a video that was prepared by a group of PhD students from a very close collaborative group in the Netherlands. This is a PhD student that won a particular, I think was a Nature Award for communication in science. And I shut up for 3 minutes and allow you to see how what I'm saying can be done.
I'm a scat man.
Everybody stutters one way or the other, so check out my message to you. As a matter of fact, I don't let nothing hold you back. If the scat man can do it, so can you. Everybody's saying that the scat man stutters but does and never stutter when he sings. But what you don't know, I'm gonna tell you right now that the stutter and the scat is the same thing. Yo, I'm the scat man. Where's the scat man? I'm the scat man. Why should we be pleasing any politician reasons who would try to change the seasons if they could? The state of the condition insults my intuition and it only makes me crazy and hard like wood. Everybody stutters one way or the other, so check out my message to you. As a matter of fact, don't let nothing hold you back. If the scat man can do it, brother, so can you. I'm the scat man. So check out my message to you. As a matter of fact, I don't let nothing hold you back. If the scat man can do it, so can you. I hear you all ask about the meaning of scat. Well, I'm the professor and all I can tell you is why you're still sleeping, the saints are still weeping, because things you call dead haven't yet had the chance to be born. I'm the scat man. Where's the scat man? I'm a scat man. Repeat after me. It's a Scooby-Dooby melody. I'm the scat man. Sing along with me. It's a Scooby-Dooby melody. Yeah, I'm a scat man.
So I think that I hope you got the message, and I don't want to start it again or how that happened, but is quite enjoyable. But I think let's go to the what has been done in the field. So the concept of removing and pushing away, as that bloke with the green hair was able to do, can be done using splice modulation antisense oligonucleotides, and a number of antisense oligonucleotides have been developed, and I developed two in collaboration with Sarepta Therapeutics. These antisense that skip exon 51 or 53. Of course, the different antisense are necessary for different parts of the gene because not everybody has the same mutation. And targeting exon 51, for example, deal with approximately 15% of children with Duchenne, targeting exon 53 for 8%, targeting 45, another 8%. And our work with Sarepta now has been also extended by work by other companies. There is one company called NS Pharma that also is targeting some of the dystrophin exons. So the field is moving very fast. But I tell you what I did at the very beginning, but also what I did more recently, that is part of the work that led to this, the fact that I've been asked to make this presentation. So we started much before that video. That video was done in 2011, I think. And so we obtained funding with a UK consortium. You have the name of the consortium quite a while ago. And we started in the grant. We obtained a grant from the Department of Health in 2005. And we started in a measured, if you like, way to try to understand whether this idea was stupid or was possible and was safe. And we started in a minimalistic way first by taking a single injection in a single muscle, in a foot muscle, that then was biopsy after, because we felt if this is not working, or if this causes trouble, at least we will not impair function in these patients. So you have here the 7 patients that were studied in this open label study, and there is each individual has biopsy before and after, and you can appreciate that in most patients, in some patients was less than others, but in most patients, the level of dystrophin produced is pretty remarkable. So that was an encouraging thing, but clearly you're not going to go very far if you have to take a needle and inject every single muscle. And also, don't forget, these antisense do not correct their gene, but correct the way the gene is processed. So you need to administer them again and again. So that was our second step, that for which we obtained a massive funding. And this was also done in collaboration with the same company. And we did a dose escalation intravenous study where we use different doses, and eventually we arrived to an optimal tolerated dose. I'm not going to present everything because this is published work now many years ago, but we identified a dose that allow us to produce sufficient dystrophin to be potentially of clinical significance. These were weekly infusion. And then the study was also continued now in the States by other colleagues, the group of Jerry Mendel, and they were able to confirm the same finding, but also extend the duration of our studies into also clinical efficacy study. Although these were all open label studies, we deal with a relatively rare condition. Is not so easy to do randomized placebo-controlled studies, and these were studied at the very beginning. But not only therefore, they showed that it was possible to restore dystrophin, but also that if you compare to untreated patients from much clinical cohorts that lose the ability to walk in the way indicated by this blue line, it was possible to prolong the way these children are able to walk by several years, up between 3 to 5 years of improvement of the walking abilities. And this clearly, it does not stop the disease, but I think allows to buy more time. And indeed, buying more time is not only with the legs, but also with the breathing muscle, with several studies suggesting that compared to the untreated patient, the administration of these drugs lead to nearly half of the yearly respiratory decline in a way that is dependent on how many years you have been studied. So the longer duration study, I feel like you accumulate the benefit and the slowdown of the disease progression. And indeed, I don't think it's in my next slide, but I presented last week in Canada now information regarding the long-term effect of survival of this drug in the, if you like, in the real world in the States. So this led to FDA approving this drug in 2019 based on this data. And more recently, I, together with, again, a large consortium, also in collaboration with the same company called Sarepta, but as an academic consortium linking multiple countries, we built on what we have done, and this time develop another antisense that developed, instead of skipping exon 51, skip exon 53. And we did, built on the experience of the first time, we did a dose escalation study with a short placebo arm, and to see whether we could replicate some of the data from the previous study. And indeed, we were able to demonstrate that compared to before, this is an example of one patient, a muscle biopsy before, and this is on the right-hand side, the same exposure. There is no trick, the biopsy afterwards, but this is probably one of our best responders. But I tell you, as an investigator, when you look under the microscope a patient biopsy before and after, and this is what you find, and we look at all of them blinded, just to be clear. It really makes you go home very happy. We were able to demonstrate that there was therefore more protein present on Western blot. We were able to demonstrate with RNA studies that the antisense was inducing the expected exon skipping. We look at also immunocytochemical aspect to demonstrate that the dystrophin was present at the sarcolemma, as you expect. But I think for me, also very interesting, we published this maybe 6-8 months ago. We were able to demonstrate that the fibers, we measure in the muscle biopsy, not only if dystrophin is present, but if there is damage. And I do not go into the detail of how we measure this, but we were able to demonstrate that whenever there was dystrophin expression, where there was more dystrophin expression, there was much less damage in the muscle biopsy. So I think this is useful because it suggests also that this dystrophin is doing something useful for the muscle. Then very recently, this has been published just a few months ago, in the open label study, again, this, don't forget, this is a small number of studies. The number of children treated is in the range of 25, and 22 had arrived this particular time point of 3 years at the time of this data cut. So you can see a difference in the trajectory of the patient. It is not an immediate divergence, because don't forget, these are given weekly. They work little by little, increasing the level of dystrophin. But I think this is very similar to what we've demonstrated the companies demonstrated with the other antisense. We were able to demonstrate that compared to unmatched control, again, numbers are small, so p-values are not significant, although they are indicating clear trends. The number of patients who did receive treatment lost their ability to walk much less than untreated control. And the respiratory function decline is approximately half compared to the untreated control. And so this again led to the FDA to approve this drug just a few years ago. In actual fact, I made a mistake. The first drug was approved in 2016, this one in 2019. I apologize. I think that if one takes the, I need to emphasize this, I'm very proud and very excited. We haven't, and I'm also happy to say that these drugs are very well tolerated by children, apart from the fact that they require weekly intravenous infusion. However, I don't think we should get complacent. There is, divergence from the treated patient, but I think it takes many years to work. And clearly, and at the moment for EMA, where there are different weight of emphasis put in the surrogate endpoint as dystrophin as a biomarker compared to FDA, there are ongoing randomized placebo-controlled studies that will eventually satisfy the bar for EMA and MHRA approval, and we are doing all of them here at Bosch. However, we would all like to have something that works better. And this is, I'm not covering this because this is not work I'm doing as a promoter, but I'm involved in all these clinical trials that are now using the information that developed for the first generation antisense, the second generation antisense, and these are both cell-penetrating peptide and receptor-mediated antisense that are entering clinical trial as we speak. And I think I really hope that what we obtain in the first generation drug will be overtaken by these new therapies. But I want to move now into the spinal muscular atrophy, because this is another field where there is an immense amount of development. This is the most common autosomal recessive condition, lethal genetic condition affecting children. If you are in every 37, 40 people in the Caucasian population in UK are carrier for this recessive gene. And the condition is quite severe. You can see here a child who has been diagnosed in the first few months of life. You can see this is how much as the child can do. He's got a rapid breathing, mostly abdominal breathing, and it looks immobile because he is immobile, although his face looks quite interactive and so on. And these children without ventilatory support will not survive the age of 9 months, they never acquire sitting. So this is as severe as you can imagine. The molecular basis of the condition is the mutation of a gene called SMN1. These are recessive conditions, so this is a biallelic mutation, usually deletion. But I think what is interesting is that there is a sister gene called SMN2. SMN stands for spinal motor neuron survival factor. This gene is very similar to SMN1 with the exception that there is a single nucleotide variation, and this single nucleotide variation leads to an exclusion of exon 7, so a delta 7. While in 10% of the transcript, therefore, SMN2 does something identical to SMN1, but is only 10 times lower. And this is not functional. So I think this is the molecular basis of this difference, is a single nucleotide variation inside the exon. So this is a mutation that affected exonic splicing enhancer. And again, coming back to the video you saw before, that particular video was how to induce pushing out an exon, but with antisense it's possible to push in an exon that otherwise would be excluded. And I think this can be done by, this can be done by antisense oligonucleotides, and the antisense oligonucleotides, therefore, in SMN2, manage to retain exon 7 that here is in green, so that eventually the protein is very similar to what would have happened if you had SMN1. An important concept before I go into the clinical trial results that have been done also here at the Neuromuscular Hospital is to go back and explain a further step regarding the genetic of the condition, because you will see there is a concept that I haven't covered that links with clinical severity. And this is related to the fact that this is an interesting biological point as well, that in the general population, the number of SMN2 copy number is variable. And this is associated with difference in the outcome. So if you go back to this particular severe child that is here, you can almost be certain that he's got very few SMN2 copy number. But this can be one SMN2 copy number in type 1 SMA, two SMN2 copy number maximum. But if you have many, many more SMN copy number, you may have milder conditions that I do not have the time to discuss, but they may affect young people or even adults. You know, the more copy number you have, the more you are doing, the phenotype is mild. So I think you will see some result where there is the outcome of children with type 1 SMA, 2 copy number are compared to, 2 copy number are compared to the one with 3 copy number. And of course, as you can appreciate from this slide, the 3 copy number have a better possibility because many of them edge into milder phenotype. So just remember this. But again, from your perspective, SMN copy number is polymorphic in the general population and probably in us listening to this lecture. So the first studies were done in children with the severe 2 copy number type 1 spinal muscular atrophy. This is done by giving an antisense because the condition is a motor neuron disease. You need to reach out the alpha motor neuron to the spinal cord. Therefore, the antisense don't cross the blood-brain barrier. You need to give them intrathecally. That I think is not very practical, but it has the advantage that the tissue half-life is months. So they do not need to be given too frequently. After a loading dose, the antisense need to be only given 3 times a year. And the company that is called Biogen has done a number of different studies. I've done some studies in children, symptomatic children, and I'll show you some, but also done some pre-symptomatic children and other studies that I will not cover in milder forms. And, you know, we are here at the Neuromuscular Hospital, we've been involved in essentially all of these studies. So in terms of what the drug does to the typical children with type 1 SMA, well, it works. Motor response is only occurred in children who receive the drug instead of a sham procedure pseudo injection in the spinal cord, that this study had a placebo arm that was eventually interrupted in the middle of the study because it became unethical to continue with a placebo-controlled study in the severe population. And as you can see from here, not only, I remind you, 0 children with type 1 SMA achieve any of these, or essentially, this thing. Here there are 4 children in the placebo arm who have achieved the ability to roll, but full head control, even ability to roll in percentage, sitting and standing, was only possible in the treated children. So this was, at the time, really a very exciting work. And I showed you before this video, and I will show what happened to this particular infants, because this is the level of function that this particular children was able to achieve. So this is clearly totally unprecedented, and that this child is able, and I stopped I should have taken the audio off, but I think this has been possible for children in whom by this time, this child, this probably is 2.5 years, 3 years, by this time, this child should have been paralyzed in bed, tracheostomy ventilated, not talking, not standing, not sitting, and therefore this was revolutionary for many of us. But I think what is even more revolutionary, and it is here in this slide, so this is in the middle color, red and black, that hopefully you can see, is the trajectory of the treated patient, like the one I showed you, compared to the sham control. And I think if you leave the sham control even longer, they will go down. So children like the one you've seen are in the good responder here. But then they were symptomatic children. So the question is, what if you intervene earlier? And this is a study that has been done also by the same company in pre-symptomatic children. This came from newborn screening or had secondary affected children in a family. And as you can see, they have black and blue lines. And this is why I made the point of the difference between the 2 and 3 copy numbers, because the 2 copy numbers have less potential to improve compared to the 3 copy number. And I'll show you what happened to this group of children recruited into the study that is called the Nurture study. And you have in yellow the 2 copy number, in blue the 3 copy number. So don't forget the 3 copy number would be an advantage. And I think the exciting thing that sitting without support was achieved by all the 3 copy number, by the great majority of the 2 copy number, well, actually by all the copy number, but only 84% in this developmental window that is necessary. In terms of walking with assistance, let's go directly to walking alone, had been achieved by all the 3 copy number and by 88% of the 2 copy number, but 64 in the normal window. And you can start to see that even the video showed you of that good responder in the symptomatic treatment start to be very different from what could be achieved if you intervene earlier. And this is very exciting. And of course, has been now replicated in another study that has been published very recently, where we also performed here at the Tormont Street Hospital, where we used this time not antisense, but a company developed AAV gene therapy, where SMN is replaced just once with an intravenous infusion of an AAV that is given to those children, the affected children. And we did a study, and the company did a study both in pre-symptomatic children, this is called SPRINT, and in the symptomatic children. So, and I think I've started from the symptomatic children, where you see the improvement in the functional ability of these children compared to the untreated or natural history study, confirming that this AAV is also very effective in the symptomatic children. But if you move in the pre-symptomatic children, there is there have been 2 studies being published just a few weeks ago or months ago on the efficacy in children who had either 2 or 3 copy number. And again, the results are outstanding, with 79% of the children with 2 copy number that were able to stand alone, and 64% of 2 copy number were able to walk alone. And once you move in the 3 copy number, stand alone was 100% and walk alone was 93%. And at the same time, and this is coming to wrapping up this, we're also learning that I like to say there is no free lunch. There are, if you like, increasing understanding that these very powerful drugs such as AAV can induce problems. So people like me, I'm getting very friendly with many colleagues in the neurology department here, in the liver unit, in the renal unit, because a number of adverse events require careful monitoring and careful management. And I think these are probably, well, most likely related to the huge viral load that is given in order to correct this defect after systemic delivery. And this is probably my final slide to indicate that we are learning also for the unfortunate and rare children who may pass away. This is an example of a study where we have been involved for a child who died in the clinical trial, but he gave us, and he didn't die directly because of AAV, but because of, we are still treating a very severe condition. This where they're studying the symptomatic children, but gave us a new, very good highlight on what is the distribution of this AAV in the human body, and give us a lot of insight for what happens once you use this AAV. So this is my truly last slide, and this is to say that for people like me, this has been incredibly dynamic time. I think that the size of the response is starting to be transformative for these people, and clearly newborn clinic will be very welcome. I think that it is, the work is not finished. I think we will be treating these severely affected children. We do not know what happens after 10 years. I mentioned that in Duchenne, second-generation drugs are emerging. And I mentioned to you that we are learning the safety profile of this product. And I finish here to say that I done probably 2% of what I discussed, but the rest of the team I work with done the remaining 98%. So I'm really grateful for the team here at UCL and the Great Ormond Street Hospital, and I will stop sharing.
Thank you very much, Francesco, for a wonderful lunch hour talk. It's been very interesting, and we've already generated quite a few questions, and please do ask more on the Slido, and I will be able to ask Francesco. So if I can start, a question from Nadia. To what degree can this technology treat nonsense and other mutations?
So, so let's start from the exon skipping. So exon skipping can treat nonsense mutation, but they need to be in an in-frame exon. So you can push out an in-frame exon. Shafat, if you go back to the video that I showed from the colleagues from the Dutch funny video, that particular bloke who had a stop, or lady, that was an in-frame exon, and it was pushed away during the splicing process, and then you induce an in-frame deletion. So that can happen. Otherwise, these type of drugs cannot work with nonsense mutation. There are other developments for nonsense mutation, but I certainly, and I'm involved in some of these clinical trials, but I didn't touch those.
Thank you very much. Another couple of questions is on the primary endpoints. And maybe one is specifically concerning the older boys and young men in DMD. are there specific primary endpoints for these older children and young men? And do any of the clinical trials target these older population?
So the answer is yes and yes. So there are, if you like, the three main aspects of function, apart from aspects related to tolerability and so on, and also quality of life. But I think one is upper limb function, for which there are validated scale. One is respiratory function. I already indicated that this is, already showing some, like some hopeful result in people in the ambulant phase, but it would be expected this will continue in the non-ambulant phases. And cardiac function is the other one for which there is a lot of attention, of course, in that population. There are clinical trials both for AAV gene therapy now for Duchenne that I didn't discuss because we only started here in our hospital, and I'm not, if like a promoter, I'm involved in clinical trials, but in the Duchenne AAV trial, but I'm not developing them. And they will be targeting both ambulant and non-ambulant patients, as some of the antisense clinical trial will be targeting ambulant and non-ambulant clinical patients.
So if I may continue, kind of related to primary endpoints. So what I understand a bit better, neurodegenerative diseases, it's really difficult to develop the primary endpoints that are easily acceptable by the FDA and EMA. It seems to somebody who doesn't know much about neuromuscular, it seems that in neuromuscular disease, it's much simpler to do, but maybe it's not. I mean, how difficult was it to develop these endpoints that FDA could accept? And related to that, do they accept natural history studies, or do you have to do controls?
Paul, you were not going to ask me a difficult question. That was how we start. So I know they have a very good question. And firstly, I think that I should say, and this may be of interest to the audience as well. So but when I say I, this is plural I, meaning in collaboration with many other people. We have been developing some of these outcome measures. For example, the primary outcome measure that is used in all Duchenne studies for ambulant children is called the North Star. Well, it's called in this way because we develop it. We develop in collaboration with multiple stakeholders. In order for this to be accepted and approved, firstly, apart from the clinical studies that we had to do in natural history and so on and so forth, we had multiple meetings with EMA. I hosted, you can find the people who may be interested to find in PubMed some publication. We hosted and had debate on whether these were fit for purpose. And I think that it was a very useful debate because there were many others, and there were probably some we learned both. I think EMA learned from us some perspective, and patient more than us. And we learn from EMA some of their perspectives. So the clinical outcome measure are now validated, and they used both by and approved both by FDA and EMA. There is still a disconnect between role of surrogate endpoint between EMA and FDA. FDA is much more flexible. EMA is less flexible, and that is why there is, if you like, a time lag between approval in Europe and in the States. I think that we are in the answer to the final question is that, in actual fact, last week I've been in touch with EMA because we would like to arrange another meeting to understand where the field is regarding requirement for placebo-controlled study. They insist, and FDA insists, placebo-controlled study for AAV gene therapy, but any person who is sensible, starting from the families and the children, can unblind the treated child within the first 2 weeks. So I think we are pretending that we do AAV gene therapy randomized placebo-controlled study, but this is a lie. And not a lie, you know what I mean? So the regulators pretend we do it but we can unblind everybody in 90% of them the first 2 weeks. So it's not a randomized placebo control. Why don't we talk about this? But if you go as a company, it's very difficult. So we are arranging this as investigators and families to say, look, this just doesn't make sense. Can we discuss it?
Yeah. We had experience like that where you had to do LPs as a placebo control. I mean, this is ridiculous. So go to some of the other questions. There's a question regarding the inheritance and looking at DMD and SMA group of diseases. Is there an increased incidence in families from consanguineous with consanguineous marriages?
So for Duchenne, the answer is no, because it's X-linked, and therefore, the risk is the same irrespective of sex, religion, marriage, and whatever. And in actual fact, perhaps something that is important for people to realize, because there is a little of this, this has been X-linked. It looks as the fault is always with females, but actually, a significant proportion of females will get mutation also from mutated sperm cell from their father. Of course, so if I cannot have a boy with Duchenne muscular dystrophy, but my daughter potentially could be a carrier, and because it's X-linked, she will not have clinical features, and she is at risk of having a child with Duchenne. So I don't think we should only blame females. We should share the problem. This is very important to discuss with families, because otherwise you induce, some sense of guilt, and clearly nobody is at fault. But to be X-linked, I think, is a little of a misnomer, because males may be initiator as well. I think that for the autosomal recessive, I think that it is such a common disease that you find it is the most common recessive disorder, together with cystic fibrosis, in all population. And it may be it is likely that in some areas where there is consanguineous family marriages, this may be even higher than what I described. But I don't think there have been major report of huge variability that really makes such a big difference, because it is so common in the general population anyway, that if like the enrichment of an ultra rare disorder, that is what happens for consanguineous marriage, doesn't appear to really provide a major additional risk.
So I don't know if this is a difficult question or not, but how often do you see females affected due to X inactivation in that?
No, this is an easy question, because I still see patients, believe it or not, and I still remember how they look like. I was in clinic yesterday because I moved my clinic on Thursday, so I had to regain.
So for us, it's in some of our X-linked diseases. That's very common.
I know. No. I know. No. I was joking, because this last few months we had two, and I had two of these, and these follow two different routes. So the first, coming back to what I mentioned to you, that perhaps I could have a daughter who is a carrier. The great majority of these carriers will be totally clinically asymptomatic, but often the CK is a bit elevated. And the creatine kinase that indicates some level of muscle damage that doesn't lead to problems, but is sufficient to, they may have a CK of 900, and therefore they may have a transaminase that are a bit high as well, because transaminase, not only you will know it, but some of the colleagues may not know it. They don't only come from liver, but also come from muscle. So if you damage your muscle, the transaminase will be higher. So one of the, we have now a very good collaborative agreement also with King's in South London. They see there's a major liver center in the country. So they refer to us females who have elevated transaminase and then have elevated CK. So in the last few months, I've diagnosed two, of which one is a completely asymptomatic carrier, and one actually had some symptoms and had mild symptoms, and therefore we do, as you probably do as well, X inactivation studies to understand what is how skewed is the inactivation, because although the Gaussian curve suggests that the majority of people will be randomly inactivated, you still have maybe, but just by bad luck, some people who, instead of being 50-50, is 80 and 20 or 90 and 10. They're not many, but those people, those females, can develop quite substantial weakness.
Thank you very much. There is the final questions just come in, and that might be a difficult one. Oh no, there might be more questions. Okay, so here's we go. What are your hopes for the future, given the side effects of AAVs? Do you think it might make more sense to push for more focus on AONs?
Well, I think that unfortunately, these children, if you have an affected child in clinic, it's not that you can wait 10 years and say, well, we wait 10 years and then we treat with whatever is best in 10 years' time. It's like if you develop a terrible migraine, whether in 5 years' time there will be a better treatment for it is not your interest. You want the best that is available today. So I think AAV does have substantial adverse event. There have been 2 deaths in 2 different clinical trials, and this was absolutely AAV related, just to be clear. However, it's a bit too long to go into the detail of the different products that have been developed by different AAV capsid, different promoters, blah blah blah. But I think at the moment it's probably one of the most promising therapies for sure, and I think that the company Sarepta has filed for an accelerated approval, and they will knock on the door of FDA by Easter 2023, okay? So I think that the emerging data suggests that, yes, there are risks, but the risk-benefit compared to do nothing is certainly in favor, in my opinion, of AAV as they are now. But this doesn't mean that in 5 years' time, we will not have better and safer AAV than today. I think the first generation antisense, like the one we have been working with, I think they are, slow burn. They work, but they need a lot of time. I think that we hope that the next generation antisense will really push the level of dystrophy into a completely different level. However, we also need to see how safe also them will be, because they may be some safety issues, and the clinical trial just started now for this novel generation antisense. So I would think the novel generation antisense are very promising compared to the first generation antisense. The safety profile is unknown. There is more data on the clinical efficacy of AAV, more data on their toxicity, but I think that as the moment, probably the most effective way to deal with a big problem like Duchenne is probably going to be AAV gene therapy.
Thank you. So I think probably the last one, related to drug approvals, given the limitations you just mentioned, do you think the rules should change for drug approval in the case of AAVs and AONs?
I think that I think when you go and discuss with the regulators, they usually have very good arguments. And I think this is not a fight with regulators, is to get mutual education. That is also why I was just discussing that I really am keen to have meetings with regulators to discuss the shortcoming of thinking that if they receive in a paper, say this is a placebo randomized controlled trial, this is what it is. It's not. But I think let's talk about it frankly. The same for antisense oligonucleotides. There may be the possibility to have better and independent assessment. Most of the side effects are really backbone related, the type of antisense you use as opposed to specific sequence. So I think there may be a way to facilitate toxicology package once you have done a few, so that you can make a more limited toxicology package, because I would say, and that is also the other discussion that we would like to have with the regulators, that the brutal reality is that none of the adverse event, I've been involved in AV and antisense clinical trial for a number of years now, including many failed trials from other companies. So none of the severe adverse events that were noticed in my children were ever picked up by any animal species toxicology study. Were not picked up by monkey, were not picked up by, so the non-human primate were not picked up by the mouse study. So at some point we need to have an honest discussion and say, the risk is for my kids at the end of the day. This is almost irrespective of how you design your non-human primate study. So I think that, I think we should discuss risk-benefit of the studies, risk-benefit of doing the preclinical studies, but also not pretend that the toxicology package make the study necessarily safe.
So, and the final, if you were to think of the biggest challenge now in the diseases like DMD and SMA, where there are a plethora of different treatments available now, so what are the remaining, or what challenges that are ahead that you would like to overcome first and foremost?
So you promised you would not ask difficult questions. So this is not an easy answer. So I tell you, there is an easy answer for SMA. There are an easy and difficult one for each of the two. So the easy answer for SMA is that in this country, we really need to do as other 11 European countries are doing, and as the States is doing, where newborn screening is available. Because the cost of the available therapies, there are 3 available therapies for SMA, is astronomical, and the outcome is severely blunted if you treat symptomatic patients. So nobody wins, okay? And these have been all drugs that have been tested and approved for newborn screening, for newborns in other countries. So that I think is, and I'm working with another group of people to ensure this does happen. The more difficult question is that what we are observing, and this means that our work as clinician scientists, you probably, Paul, are in a similar situation, is that to make sure that expectations are right. Because paradoxically, there is a lot of unrest now in families, or especially the symptomatic ones that are treated, because they are much better this year, but they are not normal, but they want more. But this more doesn't exist today. And I think having moved the expectation upwards, everybody looks at the best possible responder YouTube video. Everybody wants to be like, I would like to be a rower as good as you are, but it's probably never going to happen. But and things like that. So there is this this connection now, and we are working very closely with the advocacy group to make sure that we gather the group and the families, and because at the moment this is leading to a lot of discontent. Not necessarily. I'm talking in general. I'm not talking personally. Just to be clear. I think for Duchenne, the most significant hurdle is the fact that, I do not know what is your weight, Paul, but say your weight was 60 kilo. Well, of this 60 kilo, 25, 30 are muscle. So the amount of tissue is huge. So targeting muscle in a safe way that is sufficiently efficient remains something that I think we are learning how is getting much better with the next generation antisense. AV are getting better, but I think this is a moving target, and I'm pretty sure that in 5 years' time, we will not be doing any of the things we do today because we will have better therapies. They are really emerging. You are aware, Paul, both in the next generation antisense and in next generation AV, what is out there, but also other means of delivering perhaps transgene without using capsid or viral capsid. And so, the field is really moving really fast. But I think in reality, we need to do what is best for the patient today, but continue to work for the next generation. That's how Formula One car go from one year to the other. And, they don't stop just because they may not be the best possible performance, because running teaches you. And I think, again, the same I know that is for you being involved in this study is firstly a privilege, but also you learn so much. So, we are in a good, we and people like me are in a good position to really understand what are the shortcomings and to deal with biological shortcomings, but also technical shortcomings.
Thanks very much, Francesco. This has been fantastic hour, your talk and all the questions. Thanks very much for your patience.
Thank you.